My name is Elijah, my partner's name is Jayden, and this is our mitosis stop motion. On the screen now is a simple pie chart of the entire eukaryotic cell cycle. The majority of the chart is occupied by interphase, a sort of preparation step as the cell approaches division, and the remainder belongs to mitosis, which can further be divided into prophase, metaphase, anaphase, telophase, and cytokinesis. This will be the focus of our video.
This step is interphase. This is where the cell spends the majority of its life. We can separate interphase into three sections, G1, S, and G2. Both G1 and G2 are growth stages. The S phase is where the chromatin in the nucleus duplicates. Note, the chromosomes in the video should be in the form of chromatin, but they are chromosomes for explaining purposes of the video. After DNA replication, there are now two sets of identical chromatin inside the nucleus. Once the G2 phase is complete, the cell is ready to enter mitosis.
After the second gap phase and DNA has been fully replicated, the cell moves on to the beginning of mitosis, marked by prophase. Some notable events that take place in this phase include chromosomes becoming visible, the nuclear envelope beginning to disappear, and centrioles, priming areas on either side of the cell to set the stage for the mitotic spindles, which will become useful in later steps of mitosis. The chromosomes have yet to assemble in a specific order, so they're kind of just there for now.
The next step of mitosis is metaphase. Now the nuclear envelope has completely disappeared. Metaphase is the stage where the chromosomes line up in the middle or equator of the cell. They are neatly lined up and attached to the spindle fibers by their centromeres.
Now that the chromosomes are lined up at the middle of the cell and spindle fibers have been properly attached to each sister chromatid, the centromeres are broken by chromosomes as they are pulled towards opposite ends of the cell. This process ensures that each daughter cell will receive an equal amount of theoretically identical DNA. It also doesn't require chromosomes to be lined up in a certain order during the metaphase, just down the middle.
Now we are in telophase. In this phase, the cell becomes larger and two nuclei start forming. Each nuclei contains an exact replica of the original parent cell's chromosomes. The nuclei are divided by a contral ring for animal cells or a cell plate for plant cells. The cell is pinched in the middle. This is called the cleavage furrow.
The parent cell is then completely split into two unconjoined daughter cells. New semi-permeable membrane or cell wall will have to be created for this to happen in animal or plant cells respectively. But now the cells are free. Whee!
Thank you for watching our video about the cell cycle and mitosis.